Alrighty, guys, let's talk about macronutrients and about lung obstruction and oxygen therapy. Welcome to our podcast. So this is part two. If you want to see part one, we did a live stream yesterday. The audio is bad, but it's gonna get better. So yeah, ChatGPT, we left off talking about type one and type two respiratory failure, and I don't understand the difference. Well, I kind of do, but yeah, talk to us about it.
All right, let's break it down. Type 1 respiratory failure is when there's low oxygen in the blood, but carbon dioxide levels are normal or low. It's usually due to issues like pneumonia or pulmonary embolism. Type 2, though, means both low oxygen and high carbon dioxide, often due to conditions like COPD or drug overdose affecting breathing.
So wait, type 2 we have low oxygen, high CO2. What did you say about type 1?
Type 1 has low oxygen but normal or low CO2. It's usually from problems like fluid in the lungs or damage to lung tissue.
Okay, so give me examples of type 1 and give me examples of type 2 then.
Sure, for type 1, think pneumonia, pulmonary edema, or pulmonary embolism. For type 2, examples include COPD, severe asthma, or a drug overdose that depresses breathing.
So why does pulmonary edema and pulmonary embolism have normal CO2 and pneumonia?
In pulmonary edema and pulmonary embolism, the primary issue is oxygen exchange. The lungs struggle to get oxygen into the blood, but CO2 can still diffuse out. In pneumonia, it's a similar situation. Lung tissue is inflamed, impairing oxygen transfer.
Interesting. How come the oxygen can go out in these three?
So CO2 diffuses more easily than oxygen because it's more soluble and moves across the lung membrane quicker. Even if oxygen exchange is impaired, CO2 can still be removed, keeping its levels normal.
Yeah, but why? Why can't it be removed? And tell me type 2 diseases now.
The lungs are more efficient at clearing CO2 than absorbing oxygen. Type 2 diseases like COPD or severe asthma cause the lungs to retain CO2 due to reduced ventilation, leading to high CO2 levels.
Doesn't pulmonary embolism reduce ventilation or pneumonia or pulmonary edema?
Pulmonary embolism can reduce blood flow, impairing gas exchange. Pneumonia and pulmonary edema affect lung tissue, making oxygen exchange harder, but still allowing CO2 removal.
How do they allow it? Why do they allow it? I don't understand.
So even with impaired oxygen exchange, CO2 removal can still happen because CO2 is more soluble and diffuses faster. The
Okay,
lung's capacity to
that makes sense. Okay. Um, uh, let's talk about nasal cannula now. So, uh, and BPAP and NIV, if you could explain us what that is. And so, do we use this in type 1 or type 2 or both?
Sure, a nasal cannula provides low flow oxygen, often used for mild hypoxia. BiPAP and NIV, non-invasive ventilation, provide more support by helping with breathing. They're typically used in type 2 failure to reduce CO2 levels. For type 1, you might start with a nasal cannula or high flow oxygen.
Why do they use them in type 2? Why not type 1 too?
In type 2, the issue is both low oxygen and high CO2, so BiPAP or NIV helps with ventilation. In type 1, it's mainly about increasing oxygen, so simpler methods like a nasal cannula or mask usually work.
So I didn't understand how come BiPAP NIV is only for. Why don't we use it for type 1?
BiPAP and NIV focus on assisting ventilation, which is crucial for reducing CO2. In type 1, the main problem is oxygenation, so simpler methods usually suffice.
So it's
But if
diffusion the problem with type 1.
sure, in type 1 respiratory failure, the main issue is low oxygen levels
Fair.
due to problems.
I mean, I'm asking, is the type 1 problem mainly diffusion?
Got it. Yes, type 1 often involves impaired oxygen diffusion across the alveoli,
Mm,
like in pulmonary
okay,
edema
that makes
or
sense. So can we talk about stenting with positive airway pressure. And can you explain to us what is PEEP, positive and expiratory pressure? Um, yeah.
Sure, stenting with positive airway pressure means keeping airways open using pressure. PEEP, or positive end expiratory pressure, prevents airway collapse by maintaining pressure in the lungs at the end of exhalation, improving oxygenation.
Tell me more about it.
PEEP works by applying a small amount of pressure during exhalation, which keeps alveoli open. This improves gas exchange, prevents alveolar collapse, and enhances oxygenation, especially in conditions like ARDS or severe pneumonia.
What is this PEEP?
PEEP stands for positive end expiratory pressure. It's a technique used in ventilation to keep airways and alveoli open by maintaining a baseline pressure in the lungs, which helps improve oxygenation.
Positive pressure. I don't understand. Can you explain me again?
Of course, positive end expiratory pressure means that after you breathe out, a bit of pressure is kept in the lungs.
Okay,
This
good. And why do we need this?
we need PEEP to prevent alveoli from collapsing, especially in conditions where lung function is compromised. This improves oxygenation and makes breathing easier.
Okay, got it. Let's talk about CPAP now and how the recruitment of airways and alveoli.
Sure, CPAP, or continuous positive airway pressure, provides a constant level of pressure to keep airways open. This helps recruit or reopen collapsed alveoli, improving oxygenation and reducing the work of breathing.
Yeah, I get that, to be fair. So, okay, here's a question. We have obstructive sleep apnea. I've heard similar things to, like, we have similar diseases to obstructive sleep apnea. Conditions, could you talk to me about that?
Sure, obstructive sleep apnea is when the airway collapses during sleep, causing pauses in breathing. Similar conditions include central sleep apnea, where the brain doesn't send signals to breathe, and upper airway resistance syndrome, which involves partial airway collapse.
Mm-hmm. All right, let's go back to macronutrients. I want to do a bit of an overview of digestion and absorption.
All right, so macronutrients, carbs, proteins, and fats are broken down in the digestive system. Carbs turn into sugars, proteins into amino acids, and fats into fatty acids and glycerol, all absorbed in the small intestine.
Okay, can we talk about adaptations to GI tract and how they facilitate different functions?
Absolutely. The GI tract has specialized adaptations. The stomach's acidic environment aids in protein digestion. The small intestine's villi increase surface area for nutrient absorption. And the large intestine absorbs water, forming stool. Each part has unique rules.
Interesting. Remind me what peristalsis is?
Peristalsis is the wave-like muscle contractions that move food through the digestive tract. It ensures food travels from the esophagus to the stomach and through the intestines.
Mm-hmm.
It's a pretty cool process, right? Anything else you'd like to dive into?
It is pretty cool.
Glad you think so. Where to next?
Remind me the sequence of sphincters too.
Sure, the GI tract has several sphincters. Starting from the top, the upper esophageal sphincter, the lower esophageal sphincter, the pyloric sphincter, the ileocecal valve, and finally the anal sphincters.
Remind me
Each can
this. Okay, we have upper esophageal and then lower esophageal.
Yes, exactly. The upper esophageal sphincter is at the throat, and the lower esophageal sphincter is where the esophagus meets the stomach.
And then
Next is the pyloric sphincter, which
remind me
controls
what
food.
pyloric means and its anatomical location.
Pyloric refers to the region between the stomach and the small intestine. The pyloric sphincter is the muscle that regulates the passage of partially digested food from the stomach into the duodenum.
Uh-huh. And the duodenum, jejunum, and ileum are part of the small intestine.
Exactly. The small intestine has three parts. The
Why we
duodenum,
call it why we call it pyloric?
pyloric, comes from the Greek word pyloros, meaning gatekeeper, because
All
it
right.
acts as a gate.
And then we have the large intestine, and we have ascending transverse colon and descending.
Exactly. The large intestine includes the ascending colon, transverse colon, and descending colon, ending with the rectum and anus.
All right. Can you explain to me the parasympathetic nervous system's function for a well-working GI?
Sure. The parasympathetic nervous system promotes digestion by increasing gut motility and enzyme secretion. It's often called the rest and digest system because it helps the GI tract function smoothly.
Yeah, go to the specifics, man. Go to the specifics.
All right. Specifically, the parasympathetic system stimulates saliva production, relaxes the lower esophageal sphincter, increases peristalsis, and stimulates digestive secretions,
Mm-hmm.
all essential.
Can we talk a bit about the brain and buccal cavity? Tell us why it's called buccal. I don't even know the word, to be honest, guys. And how it connects to macronutrients and yeah.
Sure, the buccal cavity is just another term for the mouth. It's where digestion begins. The mouth breaks down food mechanically with chewing and chemically with saliva, which contains enzymes. This is the first step in breaking down macronutrients like carbohydrates. Pretty cool, right?
Yes, sir. So we have amylase, which breaks down what?
That's right. Amylase is an enzyme in saliva that starts breaking down starches into simpler sugars. So, when you're chewing bread or pasta, amylase is already at work.
Interesting. Let's talk a bit about fat digestion. We'll get into protein digestion too. Let's start with fat. So we have gastric lipase in the stomach, secreted by gastric cells. Could you explain us the process?
Sure, in the stomach, gastric lipase starts breaking down fats into smaller components. It works best in the acidic environment of the stomach, targeting triglycerides, which are the main type of fat. This process prepares fats for further digestion in the small intestine.
So gastric lipase is secreted by the gastric cells in the fundic mucosa. What's fundic?
Fundic refers to the fundus, which is the upper part of the stomach. The fundic mucosa contains cells that produce gastric juices, including gastric lipase. This enzyme helps start breaking down fats before they move on to the small intestine for further digestion.
Well, besides what can you give me a etymology of the word fundic, and also besides fundic mucosa, do we have other types?
Sure, fundic comes from fundus, which is Latin for bottom or base, though in anatomy it means the upper part. Besides fundic mucosa, there's also the antral mucosa, found in the lower part of the stomach, which mainly secretes mucus and hormones like gastrin.
Huh.
Exactly. The stomach's different regions have specialized cells and functions, all working together to break down food.
Interesting. Okay, and then so so how
And then,
does this enzyme require bile acids?
yes, bile acids are crucial. They emulsify fats in the small intestine, breaking them into tiny droplets, which makes it easier for enzymes like pancreatic
Wait,
lipase
no,
to
no.
further digest.
I mean, does gastric lipase require bile acid?
Ah, got it. Gastric lipase doesn't require bile acids. It functions in the stomach where bile isn't present. Bile acids come into play later in the small intestine to assist other enzymes with fat digestion.
Okay, let's talk a bit about protein digestion then.
All right, protein digestion starts in the stomach with pepsin, an enzyme that breaks down proteins into smaller peptides. Pepsin works best in the acidic environment created by stomach acid.
And
It's the first.
pepsin is made from pepsinogen.
Exactly. Pepsinogen is an inactive precursor released by stomach cells. It's converted into active pepsin when it encounters stomach acid, ensuring digestion begins only in the stomach.
Interesting.
It is. The body's full of these clever mechanisms to make sure everything happens in the right place at the right time. Sure. After pepsinogen converts to pepsin in the presence of HCl, pepsin starts breaking down proteins into peptides. Gastric juices, including HCl, create the acidic environment needed for pepsin to work. Muscular.
Go again, don't worry.
Sure. So pepsinogen is an inactive enzyme precursor. When it encounters hydrochloric acid, or HCl, in the stomach, it transforms into pepsin, the active enzyme. Pepsin then breaks down proteins into smaller peptides. The stomach's gastric juices, which contain HCl, create an acidic environment that helps digestion. Meanwhile, peristalsis, which is the wave-like muscular contractions of the stomach, helps mix the food with these juices, aiding in digestion.
Hmm. So, all the time I learn about what gallbladder is and then I forget it.
No worries, we can break it down. The gallbladder is a small organ located beneath the liver. Its main job is to store and concentrate bile, which is a digestive fluid produced by the liver. When you eat fatty
And
foods,
the
the
liver
gallbladder
is
releases bile
and
into the small.
and the liver is on our right side.
Exactly. The liver is on the right side of your body, just under the rib cage. It's a large, reddish brown organ. The gallbladder sits just below it, tucked in a little pocket. Both work together to aid digestion and metabolism.
And so, what is the function of gallbladder?
The gallbladder's main function is to store and concentrate bile, which the liver produces. When you eat, especially fatty foods, the gallbladder releases bile into the small intestine to help break down and digest fats.
Uh-huh.
So
Got it.
it's
Okay.
pretty
What do we mean by molecules being amphipathic? You have to give us the etymology. It sounds like a beautiful word. Yeah.
amphipathic is indeed a fascinating term. It comes from the Greek words amphi, meaning both, and pathos, meaning feeling or suffering. So it describes molecules that have both hydrophilic, water-loving, and hydrophobic, water-fearing parts. This dual nature allows them to interact with both water and lipid environments, like in cell membranes.
Interesting. Okay, let's talk a bit about the pancreas. So is it on the left or the right?
The pancreas is located more towards the left side of the body, behind the stomach. It's a long, flat gland that stretches across the abdomen. The
So
head
the
of the
so
pancreas.
the pancreas is posterior of the stomach.
Yes, exactly. The pancreas is located behind the stomach in what's called the retroperitoneal space. This position allows it to play its crucial role in releasing digestive enzymes into the small intestine and regulating blood sugar by secreting insulin and glucagon.
Interesting. Okay, so what's pancreatic lipase and what's procholipase?
Pancreatic lipase is an enzyme produced by the pancreas that helps break down dietary fats into smaller molecules, like fatty acids and glycerol, making them easier to absorb. Procholipase, on the other hand, is an inactive precursor that gets activated into colipase in the intestine. Colipase works alongside lipase to enhance its activity, especially in the presence of bile.
Okay.
Glad you're finding it interesting. Anything else you'd like to know about the pancreas?
Uh, well, I don't understand. So procholipase is activated in the intestinal lumen by trypsin to form colipase. I don't understand this process.
Sure, let's break it down. Procholipase is initially inactive. When it enters the small intestine, an enzyme called trypsin, which is also from the pancreas, activates it by cleaving a specific part of the molecule. This activation converts procholipase into colipase, which then helps lipase function more effectively by anchoring it to fat droplets, especially in the presence of bile.
Interesting. And Colipase stabilizes pancreatic lipase, increasing its efficiency.
Exactly. Colipase binds to the fat droplets and helps anchor pancreatic lipase to them. This stabilizes the lipase, allowing it to efficiently break down fats into fatty acids and glycerol, especially in the presence of bile salts.
Amazing. Okay. So, is it when we eat that this happens?
Yes, exactly. When you eat, especially meals containing fats, your body signals the pancreas to release its digestive enzymes, including lipase and procolipase.
All right, let's
These
go back to resistance, obstruction, oxygen therapy, and all of that. So, can we talk a bit about uh, obstructive sleep apnea treatment? So, do we want CPAP or BPAP?
Sure, for obstructive sleep apnea, CPAP and BiPAP are both common treatments. CPAP, or continuous positive airway pressure, provides a steady, continuous stream of air to keep your airway open while you sleep. BiPAP, or bilevel positive airway pressure, provides two levels of pressure, a higher one when you inhale and a lower one when you exhale. The choice,
Interesting. Okay, sorry, tell me about the choice, yeah?
no worries. The choice between CPAP and BiPAP often depends on the severity of your sleep apnea and how comfortable you are with each device. CPAP is usually the first choice because it's simpler and effective for most people. BiPAP
Mhm.
might be
Okay, let's talk about surface tension of alveolus.
sure. Surface tension in the alveoli is caused by a thin layer of fluid lining them. This tension tends to make the alveoli collapse, which would make breathing difficult. To counteract this, the lungs produce a substance called surfactant. Surfactant reduces surface tension, helping the alveoli stay open and making it easier for you to breathe.
The surfactant is outside of the alveoli then, and what it kind of makes the for sleep like as a tangent.
Actually, surfactant is inside the alveoli, coating their inner surface. It reduces the surface tension of the fluid lining by disrupting the cohesive forces between water molecules. This
Mm,
keeps the alveoli right.
interesting.
So, exactly. This flexibility is crucial for efficient gas exchange, allowing oxygen to enter the bloodstream and carbon dioxide to be expelled.
So type 2 alveolar cells extract fatty acids from the blood and synthesize surfactant. Could you talk more about that?
Sure, type 2 alveolar cells, also called pneumocytes, are specialized cells in the alveoli. They take fatty acids from the bloodstream and use them to produce surfactant. This surfactant is stored in lamellar bodies within the cells, and then it's secreted into the alveoli, reducing surface tension and helping keep the alveoli open
I don't
for gas exchange.
understand these type 2 alveolar cells. Could you talk more about them to me? Do we have type 3 and type 4 and type 1 too?
Of course, type 2 alveolar cells are responsible for producing and secreting surfactant, and they also help repair the alveolar lining if it's damaged. They're cuboidal in shape and more numerous than type 1 cells. Type 1 alveolar cells are thin and flat, making up the majority of the alveolar surface area, and they're crucial for gas exchange. There's no type 3 or 4, just types 1 and 2.
Tell me more about this.
Sure, type 1 alveolar cells, or pneumocytes, cover about 95% of the alveolar surface. They're extremely thin, which helps facilitate the rapid exchange of oxygen and carbon dioxide between the alveoli and blood. Type 2 cells are more cuboidal and are crucial for producing surfactant, which reduces surface tension. Together, these cells maintain the structure and function of the alveoli, ensuring efficient breathing. Sure, alveolar cells or pneumocytes come in a few types. Type 1 cells are thin, covering most of the alveolar surface, facilitating gas exchange. Type 2 cells produce surfactant, which reduces surface tension, preventing alveolar collapse. They also help repair the epithelium. Surfactant synthesis involves extracting fatty acids from blood to produce phospholipids and proteins, which are crucial for lung function.
Tell me again, I don't understand the different types.
Absolutely. Type 1 cells are mainly for gas exchange. Think of them as the thin, delicate cells covering about 95% of the alveolar surface. They're optimized for letting oxygen and carbon dioxide pass through. Type 2 cells, meanwhile, are smaller but vital for producing surfactant, a substance that reduces surface tension, keeping the alveoli from collapsing. They also help repair lung tissue.
Interesting. And what about DPPC?
DPPC, or dipalmitoyl phosphatidylcholine, is a key component of surfactant. It helps reduce surface tension in the alveoli, making it easier for them to expand during breathing. This phospholipid is crucial for keeping the lungs stable and preventing collapse at the end of exhalation.
I don't understand what this is.
No worries. Think of DPPC as a special molecule in lung surfactant. It acts like a lubricant, reducing the stickiness of the alveoli so they can open easily when you breathe in. This helps keep your lungs from collapsing.
Okay. Um, can I ask about infant respiratory distress syndrome, please?
Sure, infant respiratory distress syndrome, or IRDS, happens mostly in premature babies. It's caused by a lack of surfactant, which makes it hard for their lungs to stay open. This can lead to breathing difficulties right after birth. Treatment often involves surfactant replacement therapy and supportive care like oxygen.
Interesting.
It really is. It's fascinating how crucial surfactant is for newborns, especially preemies.
Um, okay.
Anything? I'm all ears.
So when lung is inflated, surfactant has minimal effect, and I don't understand why.
Great question. When the lungs are fully inflated, the alveoli are stretched, so the surface tension is naturally lower. Surfactant has a bigger role when the alveoli are smaller or collapsing, like during exhalation, because it reduces the surface tension and helps keep them open.
Tell me more about this.
Sure, surfactant molecules reduce surface tension more effectively at lower lung volumes. When alveoli shrink, surfactant concentration increases, preventing collapse. As the lungs inflate, surfactant spreads thinly, and the need for surface tension reduction diminishes. This dynamic
Dude, I
behavior
don't
keeps
understand. I can't lie.
no worries. In simple terms, surfactant works best when the alveoli are small, like during exhalation, to keep them open. When you inhale and the lungs expand, surfactant isn't as crucial because the surface tension is naturally lower.
Let's talk about the pancreas and pancreatic alpha amylase. How do these help with carbohydrate digestion? And do we have pancreatic beta amylase too?
Sure, the pancreas plays a key role in digestion, especially with carbohydrates. Pancreatic alpha amylase is an enzyme that breaks down complex carbohydrates like starch into simpler sugars, which are easier for the body to absorb. There's no pancreatic beta amylase in humans. Beta amylase is mainly found in plants and some microbes.
So pancreatic alpha amylase helps us break down carbs.
Exactly. It converts starches into smaller sugar molecules like maltose and dextrins, making them ready for further digestion and absorption in the small intestine.
And
This
starch
process,
is made out of amylose and amylopectin. What are these? What is their differences?
great question. Amylose and amylopectin are both polysaccharides found in starch. Amylose has a linear structure, making it less soluble in water. On the other hand, amylopectin is branched, which makes it more soluble and easier to digest. Both contribute to the texture and digestibility of starchy foods.
Interesting. Okay, and what about exopeptidases and endopeptidases? What are these about?
Sure, exopeptidases and endopeptidases are enzymes that break down proteins. Endopeptidases, like pepsin or trypsin, cut peptide bonds within the protein chain. Exopeptidases, such as carboxypeptidase, cleave off amino acids from the ends. Together, They help fully break down proteins into amino acids for absorption.
What's the difference between endopeptidases and exo?
The main difference is where they act on the protein chain. Endopeptidases cleave bonds within the protein, breaking it into smaller fragments. Exopeptidases, on the other hand, remove single amino acids from the ends of the protein chain. So endopeptidases do the initial cutting, and exopeptidases finish the job by trimming the ends.
I don't understand.
It can be, but you're doing great breaking it down piece by piece. Once you get the hang of these concepts, it all starts to fit together.
Tell me again.
Sure, endopeptidases break peptide bonds inside a protein chain, while exopeptidases remove amino acids from the ends. So one works internally and the other trims
Oh,
from the
I
edges.
see. And why is it important for pancreas and protein digestion?
It's crucial because the pancreas secretes these enzymes into the small intestine, where protein digestion happens. Endopeptidases, like trypsin, start breaking proteins into smaller peptides, and exopeptidases finish the job by releasing free amino acids, which can then be absorbed.
Interesting. Okay. Can we talk a bit about the small intestine and disaccharide digestion now?
Absolutely. In the small intestine, disaccharidases like sucrase, lactase, and maltase break down disaccharides such as sucrose, lactose, and maltose into monosaccharides like glucose and fructose. These simpler sugars are then easily absorbed through the intestinal lining into the bloodstream.
And how does this disaccharide to monosaccharide digestion happen? Does it have to do with enzymes like glucoamylase or alpha dextrinase, sucrase, maltase?
Exactly. These enzymes like sucrase, maltase, and lactase are embedded in the lining of the small intestine. They break down disaccharides into monosaccharides right at the surface of the intestinal cells. Once broken down, these monosaccharides are absorbed into the bloodstream for use as energy.
Okay, so this is kind of step two. So from polysaccharides, how do we take them to become oligosaccharides and disaccharides? And then what enzymes to become monosaccharides? Huh? Excuse me, not so yeah, wait. So in digestion, how will we go from poly to oligo to disaccharides to monosaccharides? I got a bit confused here because I said protein. Sorry.
No worries. So, in digestion, carbohydrates are broken down step by step. It starts with polysaccharides like starch, which are long chains of sugar molecules. These get broken down into shorter chains called oligosaccharides. Then, enzymes break those down further into disaccharides like maltose. Finally, these are split into monosaccharides, like glucose, which your body can absorb. Each step involves specific enzymes that target those bonds.
Okay, so what is CHO and why is its absorption limited to glucose, galactose, and fructose?
So CHO stands for carbohydrates, which get digested into monosaccharides like glucose, galactose, and fructose. These are the simplest forms and can be absorbed by the small intestine. Each type of monosaccharide uses a specific transporter to enter the bloodstream. The body prefers glucose, but it also absorbs galactose and fructose for energy.
Can we talk a bit about the GLUT transporters and explain a bit the intestinal monosaccharide transport, please?
Sure, GLUT transporters are proteins that help move glucose across cell membranes. In the intestines, the main transporter for glucose and galactose is SGLT1, which uses sodium to bring them into the cells. Then, GLUT2 transports them from the cells into the bloodstream. Fructose uses a different transporter, GLUT5, to enter the cells, and then GLUT2 to move into the blood.
hmm So let's
Does that
talk
help?
a bit about fat absorption now in the small intestine.
Absolutely. Fat absorption starts with bile salts breaking down large fat droplets into smaller ones, a process called emulsification. Then, lipases break these down into fatty acids and monoglycerides. These products form tiny structures called micelles, which bring them to the intestinal lining. There, they enter cells, get repackaged into chylomicrons, and then move into the lymphatic system before entering the bloodstream. All right, let's break it down into 5 steps. Step 1, emulsification. Bile salts break large fat droplets into smaller ones, increasing the surface area for enzyme action. Step 2, digestion. Pancreatic lipases break down triglycerides into fatty acids and monoglycerides. Step 3, micelle formation. These digestion products combine with bile salts to form micelles, which ferry them to the intestinal lining. Step 4, absorption. Micelles release fatty acids and monoglycerides, which passively diffuse across the microvillai's membrane. Step 5, reassembly and transport. Inside the cells, they're reassembled into triglycerides, packaged into chylomicrons, and enter the lymphatic system.
What are micelles? Where do they come from? The etymology?
Micelles are tiny structures formed when bile salts surround fatty acids and monoglycerides. They come from the emulsification of fats in the small intestine. The term micelle originates from the Latin word mica, meaning crumb or small particle, reflecting their tiny size.
So they break down to small particles and then they reassemble?
Exactly. Fats are broken down into small particles, fatty acids, and monoglycerides, then transported via micelles to the intestinal lining. Inside the cells, they're reassembled into triglycerides, then packaged into chylomicrons for transport.
Be confused.
No worries, it's a lot to take in. Think of it like this. Fats are broken into tiny pieces, then carried by micelles to your intestinal cells, where they're rebuilt into a form your body can transport. Each step has its own role in making fats usable for energy.
Hmm. Remind me, what's the difference between pepsin and peptidases?
Sure, pepsin is an enzyme in the stomach that breaks down proteins into smaller peptides. Pepsidase, more commonly referred to as peptidase, is a general term for enzymes that further break down peptides into amino acids. Essentially, pepsin starts the protein digestion process and peptidases finish it.
Okay. I want to ask about the colon and about digestion in the colon. Could you mention sodium, chloride, potassium, vitamins, and water and how that works and the absorption?
Sure. The colon, or large intestine, plays a crucial role in absorbing water and electrolytes. Sodium and chloride are actively absorbed, helping maintain the body's fluid balance. Potassium is also absorbed, though in smaller amounts. The colon also absorbs some vitamins, like vitamin K, produced by gut bacteria. Overall, this absorption process helps form stool and maintain electrolyte balance.
Hmm. Um, okay. Uh, so can we talk about this when it comes to sodium and hydrogen? What are these antiporters and how is the luminal membrane involved and the epithelial sodium channels? Hmm? Hmm?
Ah, got it. So the sodium hydrogen antiporter, or NHE, swaps sodium ions from the lumen into the cell in exchange for hydrogen ions. This helps create a favorable gradient for sodium absorption. Then, ENaC channels on the luminal membrane allow more sodium to flow into the cells, which is then pumped into the bloodstream, maintaining electrolyte balance.
Hmm. Okay. And what about the chloride then? How does that work?
Chloride absorption in the colon typically occurs via a couple of mechanisms. One involves chloride bicarbonate exchangers, where chloride is absorbed in exchange for bicarbonate. Another is passive diffusion, where chloride follows the electrochemical gradient created by sodium absorption. Both processes help maintain the body's electrolyte balance.
So what about potassium and vitamins?
Potassium is mainly absorbed passively based on concentration gradients. Some vitamins, like vitamin K, are produced by gut bacteria and absorbed in the colon, contributing to overall health.
Okay, now let's talk about the resistant starch, polysaccharides, and cellulose. What do these do? Like, are they digested and absorbed in the small intestine? What's going on?
Resistant starch, polysaccharides, and cellulose aren't fully digested in the small intestine. They reach the colon, where gut bacteria ferment them, producing short-chain fatty acids that support colon health. Cellulose, a type of fiber, mainly adds bulk to stool and aids in regularity. These compounds contribute to a healthy digestive system.
Hmm. So insoluble fibers make up the cellulase, uh, or no?
Exactly. Insoluble fibers like cellulose don't dissolve in water. They add bulk to stool, helping food pass through the digestive tract more easily. This promotes regular bowel movements and can prevent constipation.
Can really end this by talking about microbial fermentation.
Sure, microbial fermentation happens when gut bacteria break down fibers like resistant starch and cellulose. This process produces short-chain fatty acids, which nourish colon cells, reduce inflammation, and support gut health. It's a key part of how fiber benefits our digestive system.
And we have acetate, propionate, and butyrate. What are these?
Acetate, propionate, and butyrate are short-chain fatty acids produced during microbial fermentation of fibers. They provide energy to colon cells, support the gut lining, and help reduce inflammation, promoting overall gut health.
Interesting. Okay, guys, thank you for watching. See you at the next one.